Good evening. Thank you all for joining us tonight. My name is Emily Hensley, and I am the manager of annual leadership giving here at the Garden. And on behalf of the Garden and myself, I want to sincerely thank you for being a part of the Garden Giving Societies. We're pleased to provide our members with special programs like the one we have planned for you tonight. Your membership contribution makes everything we do at the Garden possible, including our plant science and conservation efforts around the world, with several key programs in Latin America. I know that some of you joined us for our first session on Latin America, and we're excited to take an even deeper dive this evening. Tonight, we will hear from 3 scientists working in our Latin America programs. First, I would like to introduce Dr. Monica Carlson, a plant evolutionary biologist whose research focuses on understanding and describing plant diversity in the neotropics, with special emphasis on the genus Auditorium in the aroid family. Dr. Carlson earned her bachelor's degree at the Universidad Central de Venezuela, her native country, and then came to St. Louis to pursue her graduate education, earning a master's and PhD from the University of Missouri-St. Louis. Following a postdoctoral fellowship at the Smithsonian National Museum of Natural History, Dr. Carlson returned to the Garden in 2017. She now serves as a scientist and education program manager in the Latin America department. Next, we have Dr. Julian Aguirre Santoro, a botanist and evolutionary biologist specializing in the taxonomy and evolution of plants from the American tropics, with a particular focus on the pineapple family. As the curator of vascular plants for Latin America at the Missouri Botanical Garden Herbarium, he works to make one of the most extensive collections of Latin American plants more accessible and useful through research on conservation, taxonomy, and evolution. Dr. Aguirre Santoro earned his PhD from the New York Botanical Garden and his master's degree from the National Museum of Rio de Janeiro in Brazil. Finally, we have Dr. Kelly Anderson, a tropical forest ecologist specializing in plant ecology and soil biogeochemistry. Her research focuses on plant community traits and ecosystem responses to global changes. She began her career as an assistant professor at Nanyang Technological University in Singapore and joined the Missouri Botanical Garden as a research scientist this past August. Dr. Anderson received her bachelor's degree from the University of Wisconsin and a PhD from the University of Illinois. Her interest in tropical ecology was sparked during her 10-year residency at the Smithsonian Tropical Research Institute in Panama. So please join me in welcoming Dr. Anderson, who we will hear from first.
Thanks so much, Emily, for that nice introduction. And thank you all for being here tonight. It's super exciting to be able to talk about my research to the community that is engaged with the Missouri Botanical Garden. So thank you for coming. And tonight I want to take you on a journey to the tropical forests and looking at some of the hidden diversity of tropical forest. So take a moment and think about diversity in a tropical forest. What do you think about? Perhaps you're starting to think about all of the beautiful and amazing megafauna, the things that draw us to the tropics that we want to go see. A lot of people start here and get more interested in things that support these very important animals. If you're like me, you go to the tropics to see the trees, right? These are some of the largest organisms in the world. Here I am with one of the largest diameter trees and one of the tallest trees in Southeast Asia. So we have a lot of very charismatic trees, just like we have charismatic animals in the tropics. We also have a lot of charismatic fungi, which are becoming very popular today in cultures around the world. You'll see mushroom stickers and all kinds of decorations that are associated with mushrooms. And they play a very important role in tropical forests. So, tropical forests store all of this biodiversity, and they're very important for that. But they also store a lot of carbon, and that's very important when we start to think about things like global changes. What's really interesting to me as a tropical forest ecologist is that these forests that are so biodiverse and so important for carbon storage are actually growing on some of the lowest nutrient soils in the world. And so one of the questions that we have as tropical ecologists is, how do these tropical forests maintain such high biodiversity and carbon when they're growing on very low nutrient soils? I don't know the answer to that, but we're starting to get there. So again, even as ecologists, when we think about a tropical forest, this is what we think of, right? Tropical forests are very important. We see a lot of different tree species that are represented in this image, and we think about their importance for carbon dioxide cycle or carbon cycling, right? But there's a whole world that is occurring below those trees. And this is what I mean when we're thinking about the hidden diversity. So what's happening below those trees that allow them to support such high biodiversity and carbon that's really important for global carbon cycling and storing global biodiversity. So tonight I want to introduce you to the black box of the hidden diversity, which are roots. And so I don't know if you can see the brilliant red colors of these roots, but roots come in all shapes and sizes, and they do really important functions for tropical trees. So my argument is that in order to understand how tropical forests maintain such high biodiversity, we need to better understand what's happening below ground. And integrating this below ground perspective will give us a new idea of how these forests store such high biodiversity. And a lot of people ask me, why roots? Why do we start there? There's so much to explore in the soil. Why start here? And for me, it starts because of all the really important functions of tropical roots. So if we just think about what they do, first and foremost, they keep the trees standing upright, okay? So the trees don't just fall over with the slightest breeze. Those roots are also really important in organizing the soil structure so that with all of the heavy rains in the tropical rainforest, that soil doesn't just wash away. So the roots are really important in creating this glue that maintains the soil and maintains the trees standing upright. Having these deep roots to help anchor the tree also allows the trees to get access to water, especially in times of drought. So part of global warming is not just having changes in the weather patterns or increase in CO2, it's dealing with seasonal droughts that get extended. And so having these deep roots can really be helpful during times of water stress. So those roots go down deeper in the soil where the water takes longer to evaporate, and they can take up this water to support the trees throughout dry seasons and during droughts. As I mentioned earlier, tropical forests are very important for the carbon cycle, and a lot of that carbon is stored below ground. And in fact, if we really want to understand things like carbon storage and how can we reduce the amount of carbon going into the atmosphere, I would argue that we need to start thinking below ground and all of this carbon that the roots are storing that takes longer to get into the atmosphere than things that cycle very quickly, like leaves. They decompose within a couple of months in the tropics because it's warm and wet, whereas the roots will take several years to decompose. So the carbon that the roots have stays in the system for much longer. So it's really important to understand roots, to understand carbon cycling in tropical forests. And the part that I study and think is the most interesting has to do with how plants get the nutrients, right? So I started today's talk thinking about how tropical forests have such high biodiversity. And roots are helping these trees maintain their biodiversity on these low nutrient soils. So tropical trees have a whole suite of different strategies that help them get at the nutrients in these very old weathered soils that don't have a lot of nutrients. So they're very important in nutrient cycling along with other types of cycles, such as the water cycle and the carbon cycle. And finally, the reason I show the mushrooms is that the roots are the organism, the part of the tree that's directly interacting with things like mushrooms. So mushrooms have the fruiting bodies that are beautiful, but they also have this network of hyphae throughout the soil, the leaf litter, the trees that are decomposing, and that's helping the roots get the nutrients that they need. But it also means that those roots are interacting with all of these different parts of the below ground soil community. So things like the mushrooms and their hyphae, all of the creepy crawlies that we discover when we start to dig into the soil. So things like earthworms or different types of bacteria and a whole suite of different organisms. So when people ask me why roots, because the roots are really important for tropical forests and how they function. And so I have a lab group, and our lab motto is that trees are cool below ground too. So one of the things that people often will ask me about roots and studying roots is that, if we're just paying attention to things above ground, we can see it, and it gives us an idea of how to study it. But how do you study roots if we can't even see them? And so one of my students uses a methodology that's very important, and I call it the Indiana Jones methodology, where you are searching for a hidden treasure. You start underneath a tree that you want to know something about the roots, and you just start digging until you find that root that connects directly to that tree. And then you know that those roots are from that tree, and you can start to learn something about how that tree is getting things like nutrients and water. So that for me is the fun part of collecting roots, this Indiana Jones style root tracing, but it takes a lot of time. And if we're thinking about a forest with hundreds of species of trees, it would take more than one PhD to study all of the trees in that ecosystem, that forest. So the other main methodology that we use in tropical forests to take into account all of the biodiversity above ground is just by taking cores, just taking a chunk of soil and measuring all of the roots that occur in that soil. So if you think about all of the species above ground and the big tangled mess that we're trying to walk through, it's even messier below ground. And so we're cutting into that and just getting a slice of that diversity and how the different plants above ground are being supported by all of the roots below ground. And so I will leave you with that and question you to think about the roots and what's going on below the surface. Next time you see a beautiful flower or a really cool, beautiful tree, what's happening to support those flowers and to support that tree, and thinking about the roots. Thank you. And now we can move on to the next speaker, which is Dr. Julian.
Hello, everyone. Thank you for coming. All right. So I'm going to tell you a little bit about my research. I'm Julian Aguirre Santoro. My title here at the Garden is Curator of Vascular Plants for Latin America. And people often ask me, why do you come to Missouri to study Latin American plants? And I'll tell you why, because for me, it's a privilege to be in this institution. As a Latin American botanist, we always heard of the Missouri Botanical Garden because it has been a long history of the garden and presence of the garden in Latin America. This is just a little example of all the publications that have come out of this botanical garden to understand the Latin American flora. So as you can see, I learned about botany from these books that came from this institution. So for me, it's like a dream to be here. And it's a privilege to be the curator of the Latin American collection. And this is just an example of all the resources that come from this institution to study Latin American plants. But this graph is one of the most impressive ones. As you can see, I put a lot of the institutions that hold collections from Latin America and the world. And you can see that Missouri Botanical Garden, with more than 2,500,000 specimens, is the lead institution in Latin American plants. So it's really an impressive collection of plants from the American tropics. And even if you compare, here the orange bars are the Missouri Botanical Garden, and the blue bars are the largest herbarium in each country. So, for example, in the top one is very little, but the top one is the largest herbarium in Peru. It has that amount of Peruvian plants compared to the Missouri Botanical Garden, has almost the same amount of plants than the largest herbarium in Peru. And this pattern is repeated across different countries in Latin America. So it's a very important collection still today, and I'm here to help it grow. So as a curator, if you don't understand what that means, what it means to be a curator of a collection, I have to go through all the process to make this collection grow, to enrich this collection, but also to make it available to the community, to the scientific community, but also to the community in general. So that is a process that includes from going to the field and find the plants, collect the plants, and bring all the information. During that process, train some students, pass my knowledge to other people, but also bringing the plants back to St. Louis and put them in this piece of paper, which we brought a few in the tables behind you, and also make it available through databases, and then the most important part, through knowledge. So we use this information to produce scientific literature, but all other types of knowledge. And all this information that we bring from the field and put in these pieces of cardboard is very important. This is what we use as scientists to do our analysis and do our publications. So this is an example of a herbarium sheet that shows not only the sample of the plant that we collected, but also the label information, which is a very rich information. So as you can see, it has information about the geography, so where these plants grow, information about the morphology and the ecology or biology of the plants, and also a little bit of history, who collected the plant, how many plants this person has collected by this date, and etc., etc. So if we put together 2 million plants, the richness of information is amazing. So it's my job to make this useful and available. So this is an example of how I use this information. Just by plotting the geography of each collection of these plants, sometimes I notice there are odd patterns in distribution. So I was studying this genus, Disteganthus, and I put together a lot of the localities where the plant has been collected, and I noticed that this plant in orange was odd, that it was not in the same place as the other ones. So I went there, I took a plane, found the plant, collected it, and then discovered that it was in fact a new species that we draw and then we publish in a scientific journal. Similar example here with this species of puya. I saw that most of them are here in Colombia, but then there was an odd individual down here in Ecuador. Same thing for this species, and it's the same case. And I only figured this out by looking at the collection that we have here. So there are many plants in the world. In Latin America, have thousands of different plants. So for me to focus and publish things that I can contribute really, I choose a model study. So I chose the pineapple family. I've been studying this family for more than 20 years, so I think I know them very well. And I think they're fascinating. To me, this was a perfect model to study Latin American plants, because if you can see the little map in the corner, the family is almost exclusively in the American tropics. They're not in Asia or Africa. There's only one species in Africa, but most of the diversity is here. But it's also a group of plants that are everywhere in any ecosystem that you can imagine: high elevation, low elevation, deserts, forests, etc. It has very cool adaptations. If you know bromeliads, which is the formal name of the pineapple family, some of them grow on trees, as epiphytes, but some of them grow on rocks, as you can see down here. Some of them hang on the trees like this Spanish moss. And they also have cool adaptations, like different pollinators come to visit the flowers. They absorb water from the leaves rather than the roots, which is very cool. So I found this a very interesting system to study. So from here, I have an agenda of research that's gonna keep me busy for many years. So mainly, I like to study the diversity. Oh, I don't know what this is not moving. Oh, there you go. So I like to study diversity, just learn what we have in that family and go look for new species. So I don't know what this is, a little slow. So these are examples of the genera or the groups that I have studied, and I have published manuscripts to understand better the diversity of these genera. This is a little example of new species that I have discovered. We normally have to make drawings of these plants, so we show the characteristics very well, but we also put some pictures of the plants if we see them alive. I also use this knowledge to extract DNA and reconstruct the evolutionary tree of the groups in this plant family. So this is an example of publications that I released a few years ago, trying to understand why this group of plants, how this group of plants were related to each other, and they only grow on the coasts, in the coastal parts of the continent. And then I was able to build an evolutionary tree of this group and understand a lot of questions. Recently, I published this evolutionary tree of the genus Puya, which is a genus that's mostly diversified in the Andes, in the mountains. And we were able to discover that they originated in these deep dry valleys in Bolivia and Peru, and then they moved to the high elevation ecosystems in the Puna and Paramo, where they diversify mostly. And right now I'm studying this very strange genus Grahia, which is mainly a group of plants from cloud forests in high elevation, but it's completely unknown. This is a project that I'm still working on. And I also use this information to study the conservation of these species. So who tells people if these species are in danger or not is us, the specialists in the group. So I use a lot of the information from the herbarium and my field expeditions to figure out if some of these species are in danger and risk of extinction. So I frequently meet with not only other scientists, but also environmental authorities to get together and decide if one species is in danger or not, or if we need to do something about it. So this is an example of an exercise that we did in 2020 to reassess the extinction risk of all bromeliads in Colombia after 30 years. So 30 years ago, they did it for the first time, and then we reassessed and we made some interesting, important changes that at the end was used by the Colombian government to propose new possible areas for protection on potential national parks in the country. Okay, and right now I've been here at the garden only 1 year, so I'm developing new projects. Right now I'm very interested in understanding the epiphytic plants from the Chocó forest, which is one of the rainiest places in the world, and the one that has the most epiphytes in the world. Also studying a little bit of evolution in the ecotones of drier ecosystems to very humid ecosystems in Brazil, and keep working on taxonomy of bromeliads, which is the part that I enjoy the most. So with that, I'm gonna leave you with Dr. Monica Carson. Thank you.
Thank you, Julian. And thank you, everybody, for coming today. So I am the last speaker of the night. And after I finish, we can get questions from everybody for all three of us. So my name is Monica Carson, and Emily already kind of gave away some of my slides, but that's okay. Oops, no, where are you going? Okay. Why is this? Okay. So I am a scientist and the education program manager in the Latin America department in the Science and Conservation Division of the Missouri Botanical Garden. My talk is going to flow with a lot of questions because I get these questions from people when I interact with them and when I give talk or when they see me out in the garden. I'm always here in the garden in the middle of the way, basically. So Emily already said I am from Venezuela, and I tell everybody I'm tropical. Like heat, like this, yes, this is my environment. When it's really, really cold, no, don't call me. So the other question that I get a lot, just like Julian, is like, why you coming from the tropics with all this diversity of plants do you end up in the middle of St. Louis, Missouri? So what I tell everybody is this. Like the collection of living plants between the climatron and the research greenhouses of the Missouri Botanical Garden is the biggest one in the world for the plants that I study. So it was just perfect for me to have a second home here in St. Louis, Missouri, where I can access really easily all my plants. Just a trivia question, like by now everybody knows it. The climatron and I also share a birthday. So the climatron opened its doors October 1st, 1960. Obviously, I'm not that old, but my birthday is October 1st. So we have a deeper connection, way deeper than we ever thought about it. So I had also the privilege of growing up in a country and in countries in Latin America surrounded by this huge diversity that both Julian and Kelly talked about. So I don't have to convince you that Latin American tropics are the best tropics to study and to understand what is going on with diversity, and especially with diversity of plants. So the question that I get a lot, so what specifically do you study about these plants? And I like to quote Charles Darwin because he was interested in evolution. How many different shapes, forms, colors, scents, animals, plants are there? Like, how do they evolve? How do they persist? How do they coexist with each other? How do they interact with each other? So it's a whole suite of questions that I try to address with a very specific group of plants. Just like Julian is studying bromeliads, I study something else, another group of plants, but basically the same premise, the same questions about how is this biodiversity of plants here and how it's being preserved through time. So I like to say that I study rapid tropical plant radiations. That means tons of species that are very closely related to each other and that came up in geological times in a very short period of time, about 5 to 10 million years ago. So somehow there was this explosion of these species, thousands of species, that all are coming from the Latin American tropics. So how do I study this huge diversity of things? Like Kelly said, like one PhD person or a student is not going to be enough. Well, I kind of divide it in two approaches mainly. The first one is go collect everything that I can out there in the tropics and discover these species. And once I'm discovering them, I can also inform our conservation strategies for these species that I'm discovering. And the second approach that I take is like, well, once I kind of figure out that I know what these species are, now I'm starting to ask like, questions beyond who you are. The question is, how are you related to each other in terms of plants and also in terms of ecological interactions? As a plant, am I related to a pollinator? Am I related to a disperser? Am I related to humans? Like the whole aspect of the environment that surrounds the plants. So I'm going to cover these two approaches in a little bit of more detail. But first, let me introduce you to the plants that are dear to my heart, which you probably have in your house, or you can buy them at Home Depot if you want to get a souvenir after this talk. So they're commonly called calla lilies, peace lilies. They're all related to each other. There are about 3,000 species of them in the wild. The commercial varieties are probably like 50 at the most, right? And within this huge diversity of ornamental houseplants, I study one particular group, the genus Auditorium, which is about half of the entire family. It's about 1,200 exclusively neotropical species. That means they only grow in the Latin American tropics, down from southern Mexico down to northern Argentina. That's the only place they grow. And as you can see, I tried to recreate the collage from the Charles Darwin quote, like tons of diversity in terms of flowers, in terms of colors, in terms of leaf. And on the table over there, I have 2 living plants that are from our greenhouse collections down like basically behind this building, and you can see they cannot be more different than, to each other. And we have hundreds of them in our greenhouses. So it's a really interesting group because it's not just thousands of species, it's like huge diversity of morphology within these species. Of course, I already mentioned they're super highly valuable as ornamental house plants because the leaves are amazing, but also in Hawaii, they are the second most important export commodity for the state of Hawaii, and it brings in between $5 and $7 million a year in cut flowers. My collaborators grow my anthuriums there in Hawaii, and we always partner with each other so we can inform each other about our research. So I have a deep connection to Hawaii as well, even though none of the anthuriums are native to Hawaii. Okay, so now that I introduced my plants, let's look at these 2 approaches. The first approach is collect. Go everywhere where you think these species grow and try to find them, right? And that has taken me to the most amazing tropical forest dominated by pine trees and oaks. This is just a very weird connection between temperate forests that we see here in the US, but down in southern Chiapas in Mexico. And believe me, there are anthuriums in there, and anthuriums that are not found anywhere else in the world. Obviously, the center of diversity, or one of the centers of diversity, is in the Amazon basin. I did my undergraduate thesis in this particular spot here in the Amazon River in Venezuela. In the Caribbean, even though they're little islands, they have tons of endemic species of anthuriums that are only found in this island. Once you hop to the next one, it's not found there. It's another species that takes the place of the previous species. So the Caribbean islands, especially in the higher mountains of the Caribbean islands, we also get lots of anthurium species. The other center of diversity for anthurium is the Andes. Most of my research covers the Andes in Colombia and Peru and the high elevation cloud mountains in the countries. So everywhere I go, I just collect. It doesn't matter how hard it is to get to them. Sometimes we use very different ways of approaching them, but the idea is to get to know them in the wild. So then we can bring the specimens here, and we can grow them here, and we can study them here. So obviously, when I get them here, we put them in the herbarium. The herbarium, if you have not been to it, Julian alluded to our collection being on one of the biggest one of Latin American plants. I call it a library of dead plants, right? Because that's exactly what it is. They're dead, but it's a library, and it tells us a whole history of what happened to those specimens. So I study, like for my plants, it's also one of the biggest collections in the world for the family. This combination of studying plants in the wild and studying them in the herbarium have actually led us to discover new species, just like Julian was alluding. And one of these species, Anthurium centimillesimum, actually is on your table over there. We collected it in an expedition in Ecuador between Tom Croat. I think you read the label of the specimen that Julian showed, just by chance. I didn't know he was going to show it. So Tom Croat was my mentor for my PhD, and we went to Ecuador. We collected this species. It ended up being the 6 million specimen that was mounted here at the Missouri Botanical Garden. You can see it there. It has a stamp. It says 6 million. But it's also his 100,000 collection. You saw the label that Julian had. He was already in the 70s, around 26,000. Well, in 2007, he already made it to 100,000 collections. So the name of the species, centimillesimum, alludes to being the 100,000 collection that he has ever done in his life. I think he's around 120,000 right now. So he's a super prolific collector. Being in the field with him and finding species with him is just amazing. The other thing that we get from this information about specimens is where these species grow. Nowadays, we can figure out, are those places still in there? So I work a lot with interns, and one of my intern projects last year was to figure out if these species that are endemic, that are only found in certain spots, if they are actually surrounded by good forests nowadays or not. I made a collection of Centimillesimum in 2007. There is still a good forest there for this collection, for this space, for this species to live or not? We figured out that about 24 so of 100 species that are super macro endemic, 25% of them have really no good forest around there. So this is a conservation priority for us. We can now name a species or make a priority list of species that we should go recollect, that we should see if we can transfer them to another closer area that is probably better protected. There is a lot to do. There is a lot that we can do to preserve these species now that we know that they are endangered, that the forest that is surrounding them is not in good shape. So that is it for the part of categorizing and describing biodiversity. So my last part is now asking, I would say, a deeper question, but asking more like interactive questions. How these species evolve, how they're related to each other, and how do they coexist and interact with other organisms where they are. So to do that, I using genomic tools, which means hundreds and hundreds of genes that we analyze all together to figure out how the relationships between the species are shaping up. And one very interesting result that we didn't think about it before was that actually the geography, the place where these species grow together, tells us more about their relationships than morphology. Sometimes we have a group of species that looks completely different from each other, but they all grow like in the Brazilian Atlantic rainforest. They're all close related to each other, even though they do not look alike. So that's one of the results that we have found out in our analysis of distributions of species. Species that are in the same general region tend to be more closely related to each other, even though they look very different from each other. And from these, we also discovered that the Andean cloud forest, the species that are in the Andean cloud forest, are actually 2 or 3 times more diverse than the ones that are in the Amazon rainforest or in other forest types in Central America, for example. So we have this very restricted area. So the Amazon cloud forest, as you can see in the map, let me see if I can, maybe, no, I can't. Well, as you can see in the map, the little blue spot is kind of like a very small area, right, compared to the Amazon, which is this huge swatch of purple. But there are tons more species in there. So what is keeping this diversity? How are these species interacting with each other? So the first question that came into mind was like, well, these species are actual real species. They're separated from each other. They might be next to each other, but they're doing something to keep themselves separate, right? Who can help on that? Pollinators, right? So we started smelling everything that we came across. Like our greenhouses have hundreds of species. Once a week, I went by the greenhouses and I started smelling everything that was flowering, and came up, talking with my collaborators in Hawaii also, of these ideas like maybe every single species smells slightly different. So now we're starting to collect samples, and we have the whole setup over there of our vacuum pump, how to collect samples for these flowers of anthuriums, and we can analyze them with gas chromatography mass spectrometry, GCMS, an analytical tool that tells us which ones are the chemical components of these perfumes. Maybe to my nose they're slightly different, but then I have the chemical behind it that tells me the chemicals are actually different. So this is me when we started the project. Lots of questions. It's like, what are the chemicals that are making up the scents? Are they different from each other? Are the species that exist in the same region sharing the same essence, or actually different in their scents? What are the insects that are attracted to this? So far, we have a lot of the the qualitative data, not the actual chemicals behind yet, but we're starting to get there. the thing that surprised us the most is that most people say like, oh, Auditorium species don't really smell, because the ones that we get at Home Depot, they do not smell. But the wild species, they smell, and they smell pretty good. And there are tons of different kinds of smell. So some smell like typical flowers, other ones like lemon, like the pine salt smell. If you have ever cleaned your house with pine salt, you recognize that smell, and you can smell it in the Auditorium flowers. And for some of these, we can actually know the main chemical compound that is responsible for what we think in our memory of these smells. So we can starting to tease apart the species that smell different from each other. And there are some that I used to call them smell bad, and I was told, well, they might smell bad to you, but to the insects, this is a very attractive smell, right? So to flies, to carrion beetles, to cockroaches even, those things might be very good and very nice scented to them. So I'm now calling them other scents, right? So the most common one is actually fermenting fruits, and that's something like fermenting banana that we have left in the kitchen for a long time. So most of them smell like that. But there are some very, very neat ones, like the wet dog scent. I know one species that smells like wet dog, and everybody that have smelled it, they're like, yep, that's a wet dog. So we do know certain compounds that smell like this, like the garlic, like the poop smell, and now we are starting to see patterns that differentiate these flowers that have these different smells. The smells that are sweet, they're only produced in the morning time. And they're usually associated with flowers that have very bright colors, like yellows or creams and oranges. The other scents are produced indistinctly in the morning and at night, but they're usually associated with flowers that are more dull color, like wine, purple, brownish colors. So there is definitely a component of attracting insects based on the smell, but perhaps also a component of attracting them based on the colors, because the color visions of the different insects differ, and also based on the behaviors, right? There are some insects that will only be active in the mornings, and some insects that will only be active in the nighttime. So obviously, we do not have the native pollinators here in the greenhouses, so that's as far as I can go between making connections between what we're doing as a research here in the greenhouses, but it will be super nice to go back to the field and test this hypothesis. And if we can extract the scents and figure out the chemicals that are producing the scents, test them in the field to see if they actually attract different pollinators. I also want to give a shout out right now, because I am the education program manager for the Science and Conservation Division, to all our fellows that are attending here today. Melissa is working with me from Colombia, studying the scents of Auditorium, but we also have Alejandro working with Julian on bromeliads, and we also have Ricardo over there working with Menispermaceae with Rosa. So all these fellows have come funded by us here to the garden to work on our collections, to work with our partners, with our scientists, to enhance their education and to be the next generation of us, of scientists that we have connections with in Latin America. So with that, I just want to thank everybody for coming and listen to all three of us. And if you have any questions, we're happy to answer them.